Hello, I would like first to thank Holly Bradshaw and Martin Maiden for inviting the three WHO collaborating centers for meningitis to present in this meeting the impact of the MRF meningococcal genome library on our work. I'm presenting on behalf of Dominique Cogan at the NIPH Oslo, Chin Wong at the CDC Atlanta, and my laboratory at the Institut Pasteur, Paris. Let me first remind you some of the key activities of the three WHO collaborating centers. First of all, to provide support to the WHO in meningitis surveillance and outbreak investigations, to strengthen country laboratory capacity for meningitis diagnosis, and mainly, which is relevant here today, is to provide identification and molecular characterizations of Neisseria meningitidis isolates. This is the key activity I would like to focus on today. I'll focus in my talk on Africa and mainly on the meningitis belt. In this slide is another reminder of the molecular epidemiology within the meningitis belt in the pre-genomic area in the early years of the 21st century. The scene was dominated by serogroup A, although serogroup W and serogroup X were already represented by significant proportions of the 773 isolates that were obtained from 13 sub-Saharan African countries between 2004 and 2010 and were characterized by multilocus sequence typing. So prior to the implementation of the Menaflic-VAC conjugate vaccine, serogroup A isolates were of the clonal complex ST5 with successive clonal replacements that were observed within this clonal complex and the predominance of isolates of the ST2859 since 2005. The data also showed that isolates of serogroup W belonged mainly to the clonal complex ST11, but also to the clonal complex ST175, while serogroup X isolates that emerged since 2006 belonged to the clonal complex ST181. The genomic analysis we performed later on isolates of serogroup A from the period 2000 to 2011 confirmed our view on serogroup A molecular epidemiology, but with more resolution. This analysis provided a spatio-temporal distribution of the isolates as revealed here in this phylogenetic tree, showing that in countries like Chad, ST7 isolate within the clonal complex ST5 continued to circulate. However, the ST2859 isolates that were expanding in most Western African countries, such as Mali, Togo, Niger, and Burkina Faso, have not caused major outbreaks in the region since the introduction of the Menaflic-VAC vaccine. The tools and the data that were available through the MRF meningococcal genome library allowed comparing of large collections of carriage isolates from Burkina Faso obtained between 2009 and 2012, immediately before and after the implementation of the Menaflic-VAC vaccine, that were compared to many other isolates from other areas in the world. The analysis grouped these carriage isolates from Burkina Faso in 9 clusters. I would like to draw your attention to the cluster 6 in the middle of the slide that was composed of serogroup A entirely. And 99% of those isolates were of the ST2859. These isolates were circulating in the year 2009 and 2010, but only one and divergent isolate was observed in 2012, suggesting that these isolates became rare or even absent among carriage isolates since the implementation of the Menaflic-VAC vaccine. This genomic analysis allowed to explain and to foresee the outcomes of the vaccination program by the MenC. Now we will move to serogroup WCC11 isolates. The genomic analysis of more than 700 isolates obtained between 2011 and 2016 from 11 sub-Saharan African countries showed the clustering of these isolates into different clades. Two aspects here. The first information is that the spread of these isolates between countries within this region was on the basis of different clades from the historical Hajj strain that you can see in the bottom of the tree. The second information is that the spread within the region of Central Africa seems to be on a distant clade shown in brown here within the tree. This information on the distinct isolates in Central Africa is also clear here in this study when we compared MenWCC11 isolates from the Central African Republic. These isolates were obtained in 2015 and 2016. The isolates were compared to other isolates from the other countries in the belt, to isolates belonging to the Anglo-French Hajj South African strain, and to the recent MenW isolates that derived from the South America UK strain. The data clearly show the separation of the isolates from the Central African Republic and suggesting the multifocal emergence of MenWCC11 isolates within sub-Saharan African countries. This multifocal emergence was also suggested from data obtained from Madagascar, a country outside of the meningitis belt. We compare here, as in the previous slide, the isolate from Madagascar obtained in 2015 and 2016 to the isolate from other parts of the world. As you can see, only one isolate from Madagascar was related to the Hajj strain and clustered with those Anglo-French Hajj strain, as you can see in the bottom part of the tree, and the Madagascar isolate in red, while all the other isolates from Madagascar corresponded to local isolates within the CC11 clonal complex, and you can see those isolates in the top of the tree here. We move now to serogroup C with a new clonal complex, the clonal complex ST10-217, that emerged in sub-Saharan Africa, in Nigeria and Niger since 2012, with isolates that first were grouped in one major clade with two subclades as shown in the tree to the right of this slide. The evolution of these isolates continued, as shown on the left side of the slide, with the original clade and its two subclades in Nigeria and Niger, and the apparition of a new clade in Mali since 2016. This new clonal complex, the clonal complex ST10-217, was new indeed and quite distant from other known clonal complexes circulating in Africa but also elsewhere, as shown in this tree. The next serogroup of interest today is serogroup X, with isolates of the clonal complex ST181 that also emerged in Niger in 2006, although they were present in the 1990s. Again, the genomic analysis showed that these isolates continue to evolve with the presence of two clades, an early clade in Niger and Chad, and a later clade that was mainly present in West Africa. However, both clades were observed in Niger, suggesting this country as a transmission source. The comparison with other available genomes of serogroup X showed that the emerging isolates of the clonal complex ST181 from both clades were quite distinct from other serogroup X isolates. Moreover, few allelic profiles for several genes differed between the two clades, and in particular, those genes involved in the biosynthesis of lipooligosaccharide. The last aspect I would like to mention today in terms of advantage of the MRF meningococcal genome library is the recent description of the molecular epidemiology in North Africa that showed the predominance in this region of isolates of serogroup B, but with distinct genotypes in the different countries of North Africa. The data from Morocco are shown here with the prevalence of isolates of serogroup B that were mainly of the clonal complex ST32, as shown in the tree to the left side of the slide. We further focus on these isolates of the clonal complex ST32 and compared them to other isolates of this clonal complex that share the same pore A and fit A as the isolate from Morocco. And as shown on the tree to the right side of the slide, we observe the close relationship between the isolates from Morocco that were also related to few isolates from the UK and from France, and to a lesser extent to isolates from Cuba. These data may reveal the transmission and spread pathways. All these spatiotemporal genomic networks I showed you today underline the need for new partnerships to enhance these studies. Future directions are now discussed within the Global Meningitis Genome Partnership with the aims of establishing a partnership to enable the expansion of genome collections across the world and establishing standardized common metadata collection and harmonized analysis tools. In conclusion, I hope that I presented enough data showing that the MRF meningococcal genome library provides a valuable platform for sharing genome data globally. The expansion of the MRF meningococcal genome library requires effective genomic analysis tools and large collections of isolates. The genomic data we have so far support the need for new vaccines with a wider spectrum in Africa, A, C, W, X, and Y, as well as the evaluation of the potential strain coverage of protein-based vaccines in some areas in Africa. The integration of genomic surveillance into the existing surveillance programs is critical for defeating meningitis by 2030, according to the roadmap of the WHO. The genomic surveillance needs to be expanded not only geographically for meningococci, but also to other species responsible for invasive bacterial infections, I mean Streptococcus pneumoniae, Haemophilus influenzae, and group B streptococci. Thank you for your attention.